Hello dear students, welcome back to another learning lessons. In my Physio-Pulse channels, today we will discuss about the factors affecting enzymatic activities in a simple, easy to understand way and exam oriented way. You will find the clear explanations, diagram important concepts here. Before you begin, if you find this video helpful, please like, share, subscribe and support the channel. Now let's understand about the factors affecting enzymatic activities. Actually, today we will learn about these are the factors which are involved in the enzyme activity like the temperature, water, hydrogen concentrations and concentration of substrates, concentration of enzymes, inhibitors, accumulation of end products, effect of activators and effect of light radiations. So this can be summarized in this way. We can say these are the 9 factors which are responsible for the enzyme activity. Now, if we one by one discuss about the factors, the first of all, the temperatures. As you know, the temperatures enzyme activity will be optimum at the normal temperatures, and their enzyme will have an optimum temperatures. At very low temperatures, the activity will be minimal. Increase in temperature up to the certain limit can increase the enzymatic catalytic activity. In the graph, you see here the 10-degree temperature to 20-degree, 30-degree, 40-degree, but this is the optimum temperature where the curve is bell shaped. And in this area, the enzyme activity has been maximum. So the maximum activity around about 30 to 45 degrees Celsius. This is in between this time. So beyond 45-degree enzyme activity also reduced, and I was also beyond 60 to 70-degree enzyme also denatured. So this is the important criteria to understand about the activity of temperature activity. Now, one important thing from temperature that the Q10, it is the factor by which the rate of catalysis is increased by the rise of 10 degrees centigrade. Generally, rate of reactions, most enzymes will be double at a rise of 10 degrees Celsius temperatures. Now, the factors number 2 is the water. Enzyme activity is suppressed by the absence of the water. Actually, in absence of water, enzyme activity will be nullified or decreased. In dry seeds, the enzymes are almost inactive. So this is the example, and hydration of the cells are necessary for the enzyme activity. Water provides a medium for the enzymatic reactions to take place. In many cases, the water is one of the reactant. And the number 3 is the pH. There is the actually the effect of pH. Enzyme activity or the each enzyme has an optimum pH. So both the sides with the velocity will be drastically reduced. And in here, this is the bell-shaped graph. It shows here the pH decides the change of the amino acid residues at the active sites. The net charges on the enzyme protein would be influenced by the substrate binding of the catalytic activity. So optimum pH may be vary depending on the temperature, concentration of substrate, presence of ions, etc. So example is that trypsin are the active in alkaline medium, whereas the diastase are the active in neutral medium, and pepsin shows the optimum activity of the acidic medium. So you see, these are the example of the pH where the enzyme activity can be maximum or the activity is highest in such cases. Now, number 4, concentration of substrate. What are these things? Actually, we know that enzyme binds with the substrate and to form the enzyme substrate complex, and that then it will be dissociated to form the enzyme plus product. So, increasing the substrate concentration increase the activity of enzyme until the active sites of the enzyme molecule is saturated. So, when it comes to saturation, that is the maximum velocity it goes. So, that is called the Vmax. So, after this saturation point, the rate of enzymatic reaction becomes steady. And we have seen the Michaelis-Menten equation in the hyperbolic curve, and then the addition of the substrate will not have any positive effect. So in a certain limit of extension, that the substrate concentration, if we have to increase, their enzyme activity also increase, and above this substrate concentration, if we have increased, then the enzyme activity will be the steady state. Another factor is the concentration of the enzyme. Enzymes have the extraordinary catalytic power. A small amount of the enzyme is enough for the large amount of the substrate. So increasing the concentration of the enzyme will increase the rate of reaction. If there is enough substrates, then this reaction will be increased. Increased number of the enzyme molecule will have more active sites. At high concentrations, the effect of inhibitors will be less. And number 6 factors is that is the accumulation of end products. End product accumulation retards the enzyme activity, and the active sites of enzyme becomes crowded with the products. So thus the substrate molecule will have comparatively lesser chances of the combining with the active sites. So inhibition by the end product is also regulation mechanisms such as the feedback inhibition or allosteric modulations that we will learn in the our next or later classes. Now, very important factors is called the inhibitors. Actually, inhibitor means that it can inhibit the enzyme activity. Inhibitor is the reaction that can inhibit enzyme activity, and types of inhibition depends on the nature of the inhibitor. What type of inhibitor is binding with the enzyme active sites, that can depend with the inhibition's activity. And we also classify, if you have to classify, then enzyme inhibitor can be competitive inhibitor, non-competitive inhibitor, or uncompetitive inhibitor. That we will discuss in the later class. But the inhibitors are less effective when concentration of the enzyme and substrate is higher in the medium. So this is called about the inhibitor. Actually, inhibitions details we will discuss in our later classes. And where enzyme bind with the substrate to form the ES complex, then it will dissociate E plus P. But enzyme, when bind with the inhibitor, it will form the enzyme and inhibitor complex. Now, the number eight factor is called the effect of activators. Some enzymes requires additional molecules for its optimum activity, generally known as the prosthetic group or cofactors. And this prosthetic group may be inorganic, metal ions, magnesium ion, Cu plus 2, calcium plus, sodium, potassium, etc. These are the complex organic vitamins derivative also, such as the NAD plus, FAD plus. They are also used as the additional molecules of the activators. And in the case of the allosteric enzymes, there are molecules called the allosteric modulators or effectors. The binding of these modulators to specific sites in the enzyme will cause more activated or less activated forms the enzymes, and thereby it regulates the overall enzymatic reactions. And number 9, this is the effect of light and radiations. Some enzymes are sensitive to the light, so some enzymes require the presence of the light for its catalytic activity. Example as proteolase enzymes involving the photoreaction of DNA repair. DNA repair also requires the enzymatic activity light. And usually enzymatic activity is reduced under the influence of the harmful radiation such as the X-rays, UV rays, beta rays, or gamma rays. Under these radiations, peroxides and are also informed will cause the oxidative stress. So in this way, we will discuss all about the factors which are responsible for the enzyme activity. Actually, these factors will have regulate the enzymatic kinetics. Already we discussed in the Michaelis-Menten equations, and that we already discussed here. So I think the exam purpose, what you want to know, that we have discussed in this video or in these lectures. And if you found these lectures helpful, do not forget to like, share, and subscribe to the channel. Press the notification bell so you never miss our upcoming biology, physiology, or biochemistry, or in these segments of lectures. So thank you for watching, and we'll see you in the next class.